Okay, since we have a lot that I'm going to be presenting today as part of the production release for our knowledge portals, why don't we go ahead and get started? So thank you all again for joining us today for our April 9th session of the Knowledge Portal webinar. Today I'm going to go through some events and then showcase our production release. So our first thing that I just want to highlight, we will be attending the American Diabetes Association scientific sessions this year in June. And this session is going to be in New Orleans. So if you will be in attendance, we'll be happy to see you there. As we did last year, our CMDKP is going to be a part of the NIDDK Scientific Corner, and we will be booth number 2324. And so, if you weren't able to join us at the event last year, we are, like I said, going to be a part of the Scientific Corner again this year. And here are some other organizations that will also be presenting as part of the Scientific Corner. There are several other organizations that will be a part of this as well, but they won't be in person. But if you're going to be in attendance, we'll all happily be able to communicate with you in person at our booths. We have some fun events lined up for this event that we'll be sharing once we get a little bit closer to the sessions in June. So keep an eye out for all of those to come. One thing that I'd like to highlight is a funding opportunity that may be of interest to you. And this funding opportunity is looking for pilot projects enhancing utility and usage of common fund data sets. So this is essentially, asking for help creating, demonstrate and enhancing the utility of the selected common fund data sets. And this can also go towards generating hypotheses and discoveries. And so essentially, this is going to be a 4 million program funding, which is broken down into 13 expected awards with a 200,000 award maximum. If you are interested in learning more about this funding opportunity, please feel free to scan the QR code to the right. And just note that the opportunity closes at the end of June. Another webinar series that we host as part of the Knowledge Center is the Common Fund Data Ecosystem public webinar series. And our next session will be Friday, April 24th, where you can hear from Uniprot, which is the world's leading highest quality, comprehensive, and freely accessible resource of protein sequence and functional information. We are incredibly excited to have them join, and we hope that you can join us as well. This is just a reminder that we have now moved to bi-weekly webinars instead of monthly webinars. And our next session is going to feature our other resource that we help with our production of Pinkbase. And so we are hosting quarterly webinars on this resource, which will be Thursday, April 23rd at this exact same time. And we will be covering the knowledge graphs that are available on Pinkbase. So this session might be great for you if you're, new to knowledge graphs and you're curious how they can help you with your research, or if you're someone who is a veteran of Pinkbase and you want to give some feedback, or if you have any questions and you would like to get them answered in real time, we'd be happy to have that with you. So thank you all for taking an opportunity. So I do see a note in here. How do we sign up for the Uniprot? We can send a invite through Zoom to help you register for the event. And this we can send after this session. So if you're interested in that, it'll be a registration similar to this one. And again, we're looking forward to have you all participate in that as well. Okay, and so with that, I am now going to go into some highlights from our March 2026 production release. So this production release just happened, and we want to showcase some information for you regarding new portal features, noteworthy data sets, updates to our community portals, and show some updates to another resource that's one of our flagship ones, which is the Mammalian Adipose Tissue Knowledge Portal, and then give you some sneak peek into what's to come for our June release and maybe some sneak peeks to what you'll be seeing at ADA as well. So if you would like to follow along with me, please feel free to scan the QR code and or type in cmdkp.org into your browser. Today's production release items are going to be mostly oriented to the CMDKP, but I just want to highlight that these updates are also available on the Association to Function Knowledge Portal or A2FKP and also in our community portals that, we have as well. So again, if you'd like to follow along live, feel free to scan the QR code. So on our homepage, we have added this new flagship knowledge portal section to our homepage, which basically orients you to other portals that may be of interest. This is geared towards common disease space, as you can see here. However, each homepage will orient the user to portals of interest for that specific disease area. Now I'm going to walk through some updates to our phenotype page. We're extremely excited to showcase that the bottom line method, which you have heard from us several times before and is the cornerstone of our phenotype page. This method is the meta-analysis that accounts for sample overlap. This is the first time that we've made the bottom line results accessible via download, which you can see on the bottom right here. So if you were to click on this blue download button, all of the results would be downloaded directly to your own computer. Now, as I've said, we've had this result on the portal for a while now, but you can now cite the paper, which is currently in preprint, and you can download this for specific phenotypes of interest. So if you're on the phenotype page, as I said, this is the phenotype page for coronary artery disease. You can download that specific data. Now, you might also want to bulk download this data, and this is available from programmatic access from our Human Genetics Amplifier bioindex, as you can see here. And you can explore the bioindex to see, what data you might be interested for bulk download. Scrolling down a little bit further on the phenotype page, we have another new card, and this one is representing the most significant CFDE gene sets connected to your phenotype of interest. And here again, I am showing results for the coronary artery disease. And so I wanted to highlight this specifically because this card will help you understand which gene sets are most closely related to your phenotype of interest and to also help understand the pathway of that specific phenotype. So here, the gene sets are statistically connected to the phenotype through, as I had mentioned, the Common Fund datasets, which is another example of how we have scientific interoperability between resources and incorporating data from other sites. So as you can see here, this is what's represented on the phenotype page. And before I go a little bit into this new card, I know some of you have probably heard of the Common Fund and Common Fund data ecosystem, but for those of you who are unaware, I'm going to walk through what this means and what the Knowledge Center is. So the Common Fund data ecosystem is an initiative that is part of the NIH Common Fund, as I had mentioned, and the goal is to enhance the accessibility, interoperability, and usage of data generated by these Common Fund programs. So this CFDE wheel here on the left showcases 17 Common Fund programs, which are represented on the outside in this circle. These are different programs that you can explore, and they have their individual resources along, as well as being incorporated into the greater ecosystem. And in the middle, as you can see here, there are 5 programs that are support centers to help research connect within the Common Fund data ecosystem. And we are a part of the Knowledge Center, which We are funded to integrate data and knowledge generated by these groundbreaking research programs in the Common Fund data ecosystem. And you can access our resource at cfdeknowledge.org. Now I'm going to bring us back to our phenotype page here and kind of walk through this card and basically how this can help you and your research. So this card, as I mentioned, allows users to explore which gene sets from the Common Fund data ecosystem are most relevant to your queried phenotype. And so this workflow uses a novel Bayesian method, PIGEON, which stands for priors inferred from gene annotations. And this method we have several presentations on already. If you are curious about the method in detail, you can go to our YouTube page, which has some great talks about this method specifically. But essentially, in an overview of what this method does, is it infers phenotypes, additional genes, and mechanisms that might be related to a list of genes or a gene set. And CFDE gene sets here were generated by the CFDE Data Resource Center, which is one of the five resources, the centers that I had mentioned previously. So walking through this card, as you can see here, these are the gene sets that appear, and this is the 25 most significant gene sets out of the 1,300 that we are showing. So this is paginated, and if you were to click on the Show Genes and Gene Set on this button here, this will reveal all of the genes within that specific gene set. And so this corresponding nested table here showcases the gene, as I had mentioned, the overall score, the evidence ranges, this GWAS support, and additional information that might suggest how strong the evidence is for this gene. And as you can see here, there's a lot of blue links that are within the tab. So for example, here, if you're curious and a gene stands out, so for example, this is the FLT1 gene, you can click on the gene of interest to you, and this will actually take you to the Knowledge Center to browse the correlated phenotypes for that specific gene. I'm not going to go too much into this interface itself, but I just wanted to show you the interoperability that we had between the two resources. And as I mentioned, you have this specific gene sets here. So if you were to click on a gene set of interest to you, so I'm just going to click on this top one here. Again, this directs you to the Knowledge Center where you can explore traits in which the gene set affects the genetic support. So if anyone is looking at these, on the Casey and you have any questions, please feel free to reach out to us as well, since we do have people who are working on this on our team, and we'll eventually be doing other presentations on these features as well. So with that, we are going to showcase our next new exciting resource, which is the single cell browser. And to access this, I'm going to orient you back from the cmdkp.org landing page. So basically, you'll go back to the home page, and then you'll find this under the KP Labs tab menu item at the top right. By clicking on the KP Labs, this will bring you to a drop-down menu, and you can see at the bottom you have the single cell browser. By selecting the single cell browser, this will take you to this feature. And so one thing that I would like to highlight is this is not new specifically for this release, but we haven't fully walked through this resource. So we'd like to highlight it for you today. So this single cell browser integrates human genetic tissue reference map developed using single cell RNA sequencing from projects funded by the Foundation for the National Institutes of Health, or FNIH, and also includes additional external sources as well. And so these maps have data in disease and non-disease in the AMP programs. So for this work, this is being conducted through the Galton Lab at our sister resource, the Common Metabolic Diseases Genome Atlas, and they are currently working to generate the largest integrated disease-based maps for these tissues. And as I mentioned, in addition to these data sets, we'll also include disease agnostic data sets. So you can eventually see data specifically from HubMap or HCA, for example, to create maps for both reference and disease. And this is a seed resource that we will be expanding, which I will touch a little bit about on how we plan to expand this a little bit later in this demonstration. So for, example of the adipose and pancreas, there are other resources that are dedicated specifically to exploring these tissues. But again, this is another avenue for us to have scientific interoperability with these resources to provide the largest integrated maps overall. And so, as you can see here, there are several tissues that we have represented so far. We have the artery, heart, hypothalamus, kidney, liver, muscle structure, white adipose tissue, tendon, and pancreas. And now I'm going to give you a little bit of a demonstration of how to use the single cell browser. So here you can see that this page is automatically preloaded with the artery tissue, which is highlighted in blue here on this table. So as I said, this is preloaded, but you can select any tissue of interest and just note that there's a scroll option here. So underneath here, we do have the pancreas tissue mapping here. And so you can navigate by using this table at the top. So once you have your tissue of interest, here at the top, the map will give you details about what you are looking at for the metadata information regarding the methods used and specifically, the assay involved and who generated the map. So you can see here also the species, the organ, the tissue, donors, biosamples, and samples. And so underneath this section of the map, on the top left, the first plot that we have is the cell type clustering UMAP plot. And on the right, over here, we have the expression as well. But before I go into that, I just want to note that this UMAP plot is interactive. So as you hover over the different clusters, the metadata appears in a pop-out that looks like this. So here you can see, as I mentioned, more information for the metadata specifically, and you can explore any of this information by hovering over. One thing to note, if you want to save any of this information or the images that you see as part of this UMAP, you can also download any of these images directly to your computer and the data as well. So one thing that I want to note is you can also select by specific cell subtype. And as you select, so for example, here I have selected the smooth muscle cells. You can see that this has been highlighted not only on this UMAP plot, but also on the other plots that are a part of the browser. And the other cells subtypes are grayed out in response to this. So again, the colors correspond and the other components are grayed out. As I had mentioned, the second plot on this page represents a UMAP plot of the gene expression, and this is also preloaded with a gene of interest. So for this example, we have the ITGA8 gene, and it's faint to see, but in the blue over here for the smooth muscle cells and pericytes, you can see that there is expression of this gene. The bluer the color in this UMAP plot, the higher the expression. And for here, again, if you're interested in a different gene other than the one that is preloaded, you can type in a gene of interest. Here, this is me just showing that you can type in this gene, which is ACB, and this is something of interest for this specific map as well. So if you're curious about other genes, you could also scroll on this list and select from there as well. In addition, as we had for the cell type clustering UMAP plot, this is also interactive. And again, you have the metadata as you hover over the different cells in this UMAP plot. underneath those two plots, you can actually explore traits with both stratify cell count and gene expression plots by the chosen trait across cell types. So now I'll give you an example of how you can select the traits. Oops, sorry. The first plot here on the left represents the stratify cell count by cell type. And on the right, we have the gene expression by cell type, and this is in a violin plot. This shows here, for example, that there's high expression of ITGA8 in smooth muscle cells. So as I mentioned, if you're interested in a specific trait of interest, you can use this trait selection at the top right. And so when you click on this button here, this brings a drop-down menu of the different traits you can explore. And any of the ones that are in white, you can click on, and any of the ones grayed out, you're unable to explore. So for example, here, if I were to select BMI, the page now represents all of these by BMI by cell type. And so just walking through which each graph represents, here on the top left, the cell proportion plot now represents cell proportion by BMI per cell type. And one thing that I just want to note here is each point represents the average cell proportion by cell type. On the right, we have the gene expression plot, which represents ITGA8 in this example by BMI per cell type. And again, each point represents the average gene expression per these cell types here. And one thing that I do want to note as well, these are also downloadable. So if you're interested in downloading the chart, you can do that, and all of the data will be downloaded to your laptop or computer, and the images that correspond with that. So as I had shown here, underneath, we're going to explore the top marker genes by cell type. So by scrolling down, you can explore this last plot, and this is all ranked by mean expression. So just to give some insight into how to understand this plot, the higher the mean expression, the bluer the dot will be, and the grayer the dot, and more full the dot becomes, the higher percentage of cells in this heat map here. So going into this example on this chart, we have the cell types that you can see here on this axis and the specific genes across at the top here. And so this result, if we're just looking at the smooth muscle cells at the top of this plot, this shows that the ITGA8 gene is the top marker gene in smooth muscle cells. And even though you can see it in the chart here, it's also represented in the corresponding table at the bottom as well. So this is basically two different ways that you can explore the same data. So this plot here represents the cell types, the gene, mean expression, percentage expressing, adjusted p-value, and log fold change. Another interesting result from this table shows that GHR has the highest percentage expressing cells overall. And you can see that if you were to look over here, either at the chart or the plot at the top here, this is in the adipocyte cells. If you're interested in exploring a specific cell type, this plot and table is also filterable by a cell type of interest by using this drop-down menu at the top here. You can see there's several different selections that you can pick from. And once you select one, for example, this is adipocytes, the entire plot and the table below now is showing marker genes for the adipocyte cell ranked by mean expression. And as I mentioned before, all of this work is ongoing, and so this is giving you a little preview for our June release. And Kyle Galton's team is working to have all of the tissues with integrated maps available. And we'll also be adding gene program inference using the Liger method, which is one that we are very excited to introduce to all of you. And this here is a mock-up of this feature. And so, as I said, this is a special preview. So please be on the lookout for final version, which is coming soon. If you're going to be at ADA this year, we will be showcasing this specifically there. So we're very excited to have this up and coming. And of course, we will have a, production release update that highlights, how to use this tool and how this can benefit your research moving forward. So keep an eye out for this. We are incredibly excited to have this as a future offer for you. So now I had walked through several of the new features and cards that are on our resource. I want to take a look into some of the new data sets and phenotypes that we have represented on our portals. And so, this is an exciting release, specifically, geared around the musculoskeletal and skin. As you can see here, we have several new phenotypes for both of these disease areas. We have, four of these for the musculoskeletal knowledge portal, and we have about 20-ish for the skin. And one thing that I want to note is that even though these are specifically, a part of the community portals, these are all represented on the Association to Function knowledge portal as well. So you can explore these new data and phenotypes in either the community portal that they correspond with or our other flagship portal, the A2FKP. And so, as I just gave you a preview of these data, I also want to showcase some of the work that we're doing on the community portals, in addition to the data types that we have been ingesting. And so the first that I would like to showcase is the musculoskeletal knowledge portal, which you can access at mskkp.org. Again, if you would like to look at this directly, you can put that into your browser to explore. But this is an example of what the homepage looks like. And so I'm going to go into a little bit as to why we have built this portal and how this portal can be useful to your research. So as I mentioned, why a musculoskeletal portal? So the musculoskeletal disease is common, debilitating, and costs several billion per year. And at this point in time, there's no disease-remitting treatments for osteoarthritis or age-related loss of muscle strength. And so GWAS have identified hundreds of genetic variants as associated with these musculoskeletal conditions, but there has been no clear mechanism that has come out of the research here. So for diseases with a strong genetic contribution, human genetic data enables us to understand disease, but also to identify or prioritize potential therapeutic targets. Now, before we had this portal, there was a lack of a platform for omics data in the musculoskeletal space. And there's also just limited representation for musculoskeletal in existing databases. So we put together the musculoskeletal knowledge portal to address all of these factors. The MSKKP itself is funded as an early stage knowledge base. And so our aim for this resource is to provide a comprehensive knowledge base of integrated genetic and genomic results for the musculoskeletal traits. And we are hoping that this can help establish transparent governance for the MSKKP to ensure that it's accessible to a broad array of users. And also we hope that by having a community that is involved specifically in this resource, we can have a trusted and sustainable infrastructure. And so we're very excited to have this offering to the community as well as our larger user base. So with that, our mission is to provide a single platform to integrate the growing number of valuable omics data sets that can inform musculoskeletal research, but also suggest new therapies. And so, as I mentioned, we're extremely excited to be a part of the MSKKP. And so just some details into this program. This is NIAMS funded open community web platform for musculoskeletal research. And the initial funding and continued partnership is with the International Federation of Musculoskeletal Research Societies, or IFMRS. And you can see the PIs and leadership who have helped us, build this resource to what it is today. And so going back to the homepage, as I had mentioned that, this is a genetic data mining platform aimed at accelerating discoveries for musculoskeletal traits and diseases. And right now we have integration of multi-species genetic, genomic, and multi-omic data sets. And in this resource itself, we have 577 GWAS across 606 phenotypes. And as you can see here, the user interface is pretty similar to our CMDKP and our other community portals. But the great part of this resource is there are community vetted requirements, data, and just input from the leaders in this space. And so, as our other portals have, we have interacted data analyses and visualizations. And as we continue to grow out this resource, we're hoping that, not only the community that's already involved, but individuals who, would like to participate in shaping this resource can do so. So we're constantly looking for input and feedback on these resources as well. So as I had, showcased us previously, we for the single cell browser that's on the CMDKP, we also have an MSKKP specific single cell browser. As you can see here from the screenshots, the UI is nearly identical, focusing on similar aspects of the UMAP plots and trait exploration and the heat maps to dive into this a little bit more. And so, as I had mentioned, this feature was released earlier in 2025, but this specifically, like I said, has single cell maps for the single cell RNA-seq of tendon and muscle tissues. So if you're interested in these specific maps, this is a great place for you to explore these in depth more. And so here you can have an interactive exploration of these specific MSKKP tissues. And as we're expanding on all of these, we are also going to expand for the MSKKP into additional tissues. And this is again going to be guided by expert defined standards and also for relevant tissues within the community. So I'd like to thank these developers and researchers at our labs who have helped us specifically build this browser out. And one initiative through the MSKKP that we are very eager to start to showcase here and will be a feature of one of our May webinars is our Tissue Champion initiative. So with this initiative, we have seen the challenge that with the poor data quality and lack of standards in the transcriptomic data generation and processing, this eventually leads into poor integration. And so to tackle this challenge, we have put together domain experts for 7 musculoskeletal tissues that we hope will help us define experimental and processing standards for RNA-seq data for their respective tissue. And as I had mentioned, we are going to be sharing this with the community, not only as part of our webinars, but through our resource and other guidelines that we will be creating through this initiative. And so once we have those standards built out, they will be applied to incoming data sets. And I just want to pause here for a second as well to also pitch if you are a tissue expert in musculoskeletal, we have several different, like I said, tissues that are going to be involved. We would love for you to participate in this initiative. As I said, we have identified some individuals who will be participating in this initiative, but we are constantly looking for more people to contribute. So if you're interested, please reach out to us at help@kp 4cd.org, or you can contact me directly. We are, looking to, as I said, have the community be as involved in this as they would like to be. So we're hoping that we can find some other tissue champions who are eager to participate in this as well. And if you're interested in meeting with the MSKKP team in person, there are several opportunities for you to do this. So I just wanted to highlight three conferences or congresses that you could participate if you're going to be there, where you could meet with either members of the IFMRS or our portal itself. So the first one is the IOF World Congress on Osteoporosis, and this will be coming up shortly. This is April 16th and 19th in Prague. We'll also be have representation at the European Calcified Tissue Society, or ETCS, April 24th and through 27th in Spain. So if you take time at the exhibition hall, the IFMRS will be having a booth, and we'll also have, them helping with the MacKP or MSKKP, sorry, to promote this, and they'll be available for any questions that you might have, and we can, talk to you there as well. And again, this year we'll be participating at the American Society for Bone and Mineral Research, or ASBMR, which is October 9th and 12th in Boston. And so we'll have an exhibition booth as well here where you can meet with the team. We can give personalized demonstrations. And if you have any feedback, we'd be happy to gather it from you at this as well. So there's several opportunities in the upcoming months to meet with us in person. But if you have any questions or concerns or would like personalized demonstration, we'd be happy to meet with you to go over this portal as well. Now I'm going to talk about another resource that we have for a community portal, which is the Skin Knowledge Portal, or SkinKP. And this is accessible at skinkp.org. So you can put that into your browser and you'll navigate to this homepage. Again, like the MSKKP, this user interface is similar to the knowledge portals that you know and love. And this portal is designed specifically to advance our understanding of skin disease through large-scale genomic data. And I'll tell you a little bit more about this project, why it matters, and what we're planning on accomplishing with this specific portal. So why a skin portal? So the Skin Knowledge Portal is supporting the work directly of the Skin Genetics Consortium, or SGC. And so their main aim is to, as I said, advance the understanding of the genetic underpinnings of the common skin diseases by leveraging large-scale genomic data. And this consortium basically aims to uncover the genetic factors that contribute to the onset, progression, and treatment responses of skin diseases across diverse populations. And we, the SkinKP, promote innovation in dermatology through an open, collaborative approach to discovery that is founded on genetics and encourages and facilitates open data sharing. So as I had mentioned, this is developed by the Skin Genetics Consortium and also our team at the Broad. And so I just want to take a moment to acknowledge our leadership who have been driving the development and success of this portal. So I just want to take a moment and say thank you for them for their insight into which data and information will be important to not only the skin community but those who may want to learn a little bit more about skin disease. And so, as I had mentioned, this portal, we had quite a bit of new data that is added through this production release, and so new phenotypes as well. So this portal itself has 44 genetic data sets across 28 phenotypes. And again, this is really exciting. We were able to add over 20 new phenotypes within this release alone. So you can really see how much the community input for phenotypes of interest has already been building out this specific resource. If there are GWAS or phenotypes of interest or data sets or any genomics data that you have and you'd like to contribute or you would like to see represented on this community portal, again, please feel free to reach out to us as we do want this to represent the larger skin community as a whole. And so one of our other flagship portals that I would like to share some updates on today is the Mammalian Adipose Knowledge Portal. And so some of you have maybe heard of this before. We've had several presentations and we've also introduced this at the ADA last year. And we'll also be showcasing a little bit more of at ADA this summer is again the MacKP. And so this is our homepage of the resource, which is accessible if you were to go to mackkp.org. Again, if you put this into your browser, this will take you to this specific homepage. And this portal is an open access community portal for adipose biology across multiple mammalian species. And so I'll go into this a little bit more. But this community portal, or flagship portal, as I should say, was designed specifically with adipose tissue in mind. And so adipose tissue regulates the diverse metabolic processes and plays a role in the pathophysiology of diverse phenotypes. But however, in its development and subpopulations and molecular pathways, these all seem to be poorly understood. So we hope that the Mammalian Adipose Tissue Knowledge Portal can address this challenge and look at this from several different avenues. So as you can see here, the MacKP is developed by a multidisciplinary team of researchers. And again, these are members of the leadership, and it's funded by the National Institute of Diabetes and Digestive and Kidney Diseases, or NIDDK. And so one thing that I want to highlight here back on the homepage is we have several different components of this resource that we offer, including the single cell browser, which I have mentioned now several different times through this presentation. It's also represented here, but specifically for adipose tissue. There's an expression browser, a phenotype browser, functional annotations, and a look into adipogenesis. And this resource itself has several different datasets as a whole. There are 85 datasets, and these are broken down into two different species. So you have musculus and then Homo sapiens. And so for the mouse and human, both of these are broken down by single cell and bulk RNA. And so we also, for the mouse, have a time series data as well. And so before I go into more of the data, I just want to showcase, if you scroll down at the bottom of this page, you can also explore, specific depots of interest. And here, this is just showing from Homo sapiens. If you were to select the mouse as well, a mouse diagram would appear, and you can explore the different depots. So if there's specific depots of interest, it will take you directly to those data sets of interest as well. And like our other portals, we have opportunities for collaboration, and we're also going to be, as I said, at ADA, so we'll have more information about our time at ADA in the news items, so please keep an eye out for that as we get closer to the events. And so now I want to take a little bit of time to go over the data that is in the MacKP. And so for this release, we focused quite a bit on integrating mouse data. And so as you can see here, this is our dataset browser. And this is where you can actually explore the different data sets that we have. So here it's in tabular format, where you can explore the different data set names, the species of interest, tissue, depot, etc. And so there are several different ways that you can filter on this table. So on the right, I just want to point out that each of the items that are in this table, you can filter. So again, if you're interested in a specific depot or sub-depot, that can help with the filtering as well. At the top, you could also filter by text. So if you have a data set of interest that you're already aware of, this is a great way to navigate directly to this. And so if you have a data set that is exciting to you, you can click on the Explore button here in yellow. And by clicking on the Explore button, this will take you to the single cell browser specifically for this data set. As I had mentioned, this is again the same similar user interface that I had presented as part of, the earlier presentation. And again, these are going to continue to be built out and iterated as we add in more data, and different data types. So we are very excited to, be moving forward in this specific area as well. And so, as I mentioned, this is continuously improving. So we plan to expand on the MAT-KP with the additional data sets, as I had mentioned, species. So we're looking into the addition of non-human primates, and several features. But these are some of the things that we're interested in including. So the epigenomic data, ATAC-seq, ChIP-seq, cut-and-run, transcriptomics data, single-cell RNA, bulk RNA-seq, spatial RNA, trap-seq, proteomics, metabolomics, and imaging, specifically through the lipocyte profiler, which we are working on in parallel on this. So these are just some of the ideas of where we're imagining this going, but we're also eager to hear from you. If you have any data that you would like to contribute, we are always willing to accept your data. So please feel free to reach out to us for integration of your data as well. Um, and so with that, like I said, I promised we would do a little bit of a teaser of future work, which will be for our June release. This release is tied into ADA. So if you're looking for, a firsthand experience of this new release, if you come to ADA, you'll be the first to experience all of these firsthand. So we're really excited about this. So just focusing on some of the portals that I had already touched upon today. We're for all of these going to be looking to integrate new genetic and genomics data. And as I had mentioned previously, we're going to continue to iterate the single cell mappings for all FNIH funded tissue data, and continue to ingest the additional data sets for these mappings. As I had touched upon too, the beta version of the gene program inference will be available on both the CMDKP and on MAT-KP, which we're very excited about that. Um, and in addition, on the MAT-KP will also be, as I said, including more mouse data sets. And we'll also be working on a beta mouse to human comparison visualization that we're really eager to, have as an offering on this site as well. I know I had mentioned previously for the MSKKP that will have new data ingest. We'll have the Tissue Champions initiative ongoing. We're hoping at this point we'll have, several presentations and ongoing work for those standards that we'll be able to bring back to the community. And then again, continuing with the single cell processing and pipelines for those maps. So while we have a little bit of time left, I'd love to hear from you, the community who helps, drive the new features and data and things that are on this portal that best represent your research. So what would you like to see on these portals? If you want to think about it and take some time, please feel free to contact us at help@kp4cd.org for feedback and recommendations. But before we go into the feedback session, I just want to have a large shout out to our team. Thank you so much for every single person who has put in countless time and effort towards, iterating all of these incredible portals. We wouldn't be where we are today without you. So thank you so much to our team. And as I said, we'll go back and we can kind of open this up for a little bit of discussion. What would you like to see on the portals? Is there specific data types, phenotypes, any other features that you think would be of value to the community? I'd love to pause and kind of hear from you. So if you have any thoughts or opinions, please feel free to use the chat Q&A, or if you want to raise your hand, I can take you off of mute. But we really would love to hear from you. So thanks everyone, and love to hear your feedback.
Thanks so much, Mackenzie. That was fantastic. Just to kick us off, it looks like we have a question in the chat. Someone was really interested in the Uniprots webinar that you mentioned is upcoming. How do folks sign up for that webinar or for additional webinars if they're interested?
Great. So if you're interested in attending this webinar, we will have a news item, or should already be up there already, a news item on the upcoming webinar on the CMDKP on the homepage on the news items, or you can go to the Knowledge Center, as I had shown earlier, so you can get there through cfdeknowledge.org, and there'll be a registration there. We'll also be sending this out to the mailing list. So anyone who is on the mailing list, um, if you're here, you most likely are. We can send out the link as well. And so hopefully everyone who is interested will be able to sign up through those mechanisms. And we hope to see you there. Any other feedback, thoughts, questions? We have a lot that is on our resource, and I think, sometimes there's a lot of information, so I understand if you're processing all of this. But as I mentioned, if you come up with anything, you can also email us. Okay, I'm not seeing any hands raised or any other questions in the chat or Q&A. So I will take that as people might not have any questions live, but might have some questions for us later on. So thank you all so much for joining us today. We are going to have this up on our YouTube. So if you want to share with your colleagues, please feel free to do so. And a reminder, our April second session will feature the pink graph from the Pake Base resource that we are also a part of. So we look forward to having you all there. So again, thank you all so much for joining us and hope you have a great rest of your day. Thanks, everyone.